Welcome back. So we've moved on now. We're going to look at the processes by which one cell becomes two. So this takes us all the way back to an early principle we talked about, the principle of cell theory. I remember there were three parts to that. All cells come from other cells, and that's what we're going to talk about when we're talking about mitosis and meiosis. And then the other two parts where the tissues are made from cells, and cells are the smallest unit of life. So we're focusing on here on cell division. That is the process where one cell becomes two. And so we're going to look at mitotic cell division, and then we will look at meiotic cell division. We'll start out though with a quick discussion on chromosomes and really understanding mitosis and understanding meiosis. It's critical that you understand chromosome structure and how then chromosomes behave in the cell during mitotic or meiotic cell division. So we're going to start out taking a look at some chromosomes. So before we get to look at the chromosomes, I just want to talk you through how you can actually visualize this in a microscope. This is pretty easy to do. We could do this in one of our labs on campus. So if you wanted to look at some chromosomes, what you do is you take some cells, and you can grow these cells in dishes. And so there are lots and lots of different types of cells that you can grow in dishes. Probably the most famous type of cell is a cell line called the HeLa cell line, H-E-L-A. They were isolated from an African-American woman in Philadelphia many years ago. They've been used all over the world, and her name was Henrietta Lacks, and she had a cervical cancer, and so someone isolated some cells from her cervical cancer, and so you can grow those in dishes. So you get the cells growing in a dish, and then you add a drug to those cells which causes them to divide, and what you actually do is you can actually stop them dividing, so they stop dividing in a stage of the cell cycle, a mitotic cell cycle called metaphase, and we'll take a look at that later. So what you then do is you can take those cells, or you can also take blood, and you can do this with blood as well, and that's what we're doing here. So you got these cells, whether they come from a dish or whether they've come from a biological sample like some blood. Here it's some blood, and so let's talk about the blood experiment. You take some blood, you treat it with this drug which causes the cells to stop dividing, and then you pellet the cells to the bottom of a tube in a process called centrifugation, and that's a machine which spins the tube really fast, and the dense material, the cells, contain is at the bottom of the tube, and then there's just the fluid called the supernatant is at the top, and then you dump off the supernatant, and then in the second thing here, you resuspend the cells. So I'm in my second slide here. Let's get a pen going here. You resuspend the cells in a hypotonic solution, and so that has an effect on the cells. So let's have a quick question. Why would you put the cells in a hypotonic solution? What would happen to the cells if you did that? Because if you put the cells in a hypotonic solution, think back to our discussion of osmosis, there would be more water, more free water in the hypotonic solution surrounding the cells than in the cells themselves. So water would move in, and the cells would swell and swell and swell, and you want to get them just to the point where they're about to burst. So the cells are swollen up at this point, and then what you do is you take those cells and you can drop them. You drop them about 12 inches, 1 foot, it's about 30 centimeters. You drop them onto a glass slide, and so they land on the slide, and because they're all swollen up, when they hit the slide, they burst. And as they burst, the chromosomes spread out on the slide, and so they all spread out, and they're all kind of condensed, and you can look down a microscope, and if you've got a camera attached to it, that's even better. And so if you look down the microscope, you can actually see those cells. You can see those burst open cells, won't be anything to see, but you can see the chromosomes. And then somebody who's a skilled microscopist and is used to looking at chromosomes can line all the chromosomes up. So they take the homologous pairs, that's the one from mom and one from dad, and they can line them all up so you can then see all the sets of chromosomes. And so the first thing here to talk about in chromosomes is a little bit about chromosome structure. So just now I mentioned that there are homologous chromosomes, and that's a chromosome from mom and chromosome from dad, and when they're together, then they're called a homologous pair. So they would be individually called a homologue. So in this little diagram here, we would have this chromosome would have come from mom or dad, and then this one would have come from mom or dad. And so this could be chromosome 1 from mom, chromosome 1 from dad, and so that's called a homologous pair. And so you can see all of the homologous pairs. There's chromosome 1, the homologous pairs, chromosome 2, the homologous pairs, all the way through down here, the sex chromosomes X and Y. So as what we have 22 sets of what are called autosomes. Those are the non-sex chromosomes. And then we've got an X and a Y, or you would have an X and an X. So that's what a homologous chromosome is, or that's what a pair of homologs is. But then we also have to look at a structure of a single chromosome, and single chromosomes will start out, I'll try and outline it. Here's one chromosome here, and once that's been duplicated during the cell division cycle, a second structure is formed, and that's now called a pair of sister chromatids. And we'll talk about this more when we talk about the processes of mitosis. So a couple of things to start thinking about here. There are these structures called homologous chromosomes. That's chromosome 1 from mom and chromosome 1 from dad, and then they're called a homologous pair. And each chromosome starts out as one double-stranded piece of DNA, and then before we can divide the cell, we've got to replicate the DNA, and when those two things are stuck together, that's called a pair of sister chromatids. So take a real close look at this this karyotype here. Karyotype is when you lay all the chromosomes out like this, and I want you to pay attention to the sex chromosomes down here in the bottom right, because I've got a clicker question coming up, and here it is. Okay, so that's a real quick introduction. All we're interested in to start with is a quick look at chromosomes, and what we'll start doing in a moment is looking at mitosis, and we're really going to be interested in the behavior of those chromosomes. So that's it for now. I'll see you shortly.